Hello, everyone. Thank you for joining for today's AAV workshop and roundtable discussion, a comprehensive guide to using AAV vectors in gene therapy. My name is Derek Schneeweiss, and I'll be moderating today's event. Today's workshop will be led by Dr. Andrea O'Hara, Strategic Technical Specialist at Azenta. Dr. O'Hara has over 13 years of experience in next-generation sequencing. She earned her Ph.D. from the University of North Carolina at Chapel Hill in genetics and molecular biology. Throughout the event, please feel free to send any questions you might have through the Q&A chat box located on the right side of your screen. Dr. O'Hara and our panel of experts will answer your questions during the roundtable discussion. To better understand our audience and adjust the workshop, we have 3 initial poll questions. Please reach out if you have any questions as you answer the polls. I'll go ahead and start those now. And we'll give you some time just to answer through. Okay, we'll start the next one. Okay, and then one final question. Perfect. Thank you, everyone. Without further ado, I'll hand everything over to Dr. Andrea O'Hara to begin the workshop. Andrea, can you join us now?
Here I am.
There we go. Perfect.
Hi, everybody. Thank you so much for joining us, and thank you, Derek, for that great introduction. Okay, so to get started, I just want to go through a really brief introduction. You may already know us by our new brand name, Azenta, but if you're not familiar, we are actually a union of a number of different companies, including our former name of GeneWiz. I'm just showing you here a handful of the different companies that we have. So we offer now a number of different products and solutions all under one roof. This includes really a wide variety of different options for sample exploration and sample management, including genomics and analytics, consumables and instruments, storage, automation and logistics, sample sourcing, data informatics, and then also consultative services. And those consultations actually happen across the board for all of these different functions. I'm going to be focusing heavily on the genomics and analytical services, but I'll actually be highlighting all of these different products and services throughout the presentation today. We have sites located across the globe. Our main major sites, or our main headquarters, are located within the US, EU, and in China, but we have a number of satellite locations. This is really important because depending upon where you have your samples located, you can actually submit at any number of our locations, and we can also do internal transfers as needed. So really reflecting the fact that we have a wide variety of options at your disposal depending upon where your projects are taking place. Now today we'll be discussing AAV development, and we have a number of best-in-class solutions. This includes automated solutions. These are automation for both sample storage as well as sample processing. proprietary genomics protocols, and we'll get into some more details about that in just a moment. We have a global cGMP repository and logistics. This is for your offsite storage and cold chain management. And again, we are industry experts, so we're going to actually give you a whole bunch of information today about all the different ways that we have these cutting edge technologies to really help you advance your AAV development. Now, the basic AAV development workflow is going to be divided up into 3 main stages: your discovery, preclinical, and then clinical development. And within these stages, we have a number of substages. So for basic discovery, target discovery, and then lead identification, where you're going to be creating that therapeutic. Preclinical development, where we're going to develop that model further, optimize the process or the actual therapeutic. And then finally moving into the clinical use. So clinical trials, therapeutic delivery, and then final scale up. Let's do a little bit of a deeper dive. So with our target discovery, we have a number of different products and solutions that can really assist you throughout this. Starting with our basic research, we're actually going to really just start to develop that model of the disease and screen clinical samples. And for this one, you may use your own independently gathered samples, or you may choose to outsource sample sourcing, something that we can assist you with. Now, once you have all of those samples gathered, you're going to do more to understand the disease mechanisms and identify candidate genes. Typically, you might think of doing next generation sequencing or Sanger sequencing. There's a whole other host of different tests and things you can run. And depending upon what you're doing here, we do offer consumables and instruments for doing sort of a DIY approach or doing everything in your own lab. But we also do offer a number of different outsourced options. Now, once you actually have a good idea of a candidate gene of interest, that's where you're going to be able to create that novel therapeutic. And again, we're talking about AAV here. So we're going to be talking about actually creating that gene therapy. For that, we have a number of different products and solutions, including a gene synthesis or DNA synthesis, plasmid prep, Sanger, and next generation sequencing. And then as we're going through all of this, we offer a number of storage solutions. So you might consider storing things on site at this stage. So you would just need really good, solid cryo storage. We're going to take a deeper dive looking at this create the novel therapeutic, and we're going to go through this stage in more detail step by step. So we can kind of uncover all of the different intricacies and things we need to think about when we're actually creating this novel AAV therapeutic. So here I'm showing you a basic workflow of creating this novel AAV therapeutic, and I want you guys to really take this picture in. We're going to be going through this in more detail step by step. So with AAV therapeutic design, there's a number of different challenges that we have. The first part is going to be upstream when we're actually creating these plasmids. Plasmids are really difficult to synthesize, amplify, and analyze for a number of different reasons. And then once we actually package this all together, we really need to focus on what our final recombinant AAV looks like, making sure that we have a fully intact species. We want to make sure that we have complete ITRs, that there are no point mutations, frame shifts, edits within the ITRs, but that also the transgene itself is thoroughly stable and well vetted. Now, if we go through this process, the first thing that we're going to start with here is actually creating that transgene expression cassette or creating the plasmid. Then we're going to want to confirm the sequence. We're going to want to expand the plasmid. We're going to introduce it all into the packaging cell line along with our rep cap and helper plasmid, generate those particles, and then we need to sequence what's inside of those particles to make sure that we have exactly what we want, no more, no less. So to do this, we have a number of different AAV solutions that we'll be discussing today. We're going to talk first about the synthesis options. This can be plasmid synthesis or gene synthesis. Next, we're going to talk about sequencing options, including Sanger AAV ITR sequencing. Next, we'll talk about plasmid prep and some of the things we need to consider when we're thinking about that scale up. And then finally, looking at packaged AAV sequencing, so uncovering more about what's actually inside those particles. So if we actually take all of those different graphics I just showed you here and overlay it on top of the image that we looked at previously, we're going to see here all of these different products and solutions and how they come into play within this entire workflow. So starting out again with that, synthesis option, we can either synthesize the transgene itself to go into a transfer plasmid that we've already created, or we can do plasmid synthesis and create the entire plasmid. We're going to be able to look at that sequencing of the plasmid, preparation of the plasmid, and then again, focusing on the downstream applications. We have 2 different sequencing options that we're going to be considering. So let's go ahead into that first step, which is going to be the transgene or plasmid synthesis. So like I said, to do this, we really want to think about what we are going to use and how we're going to use it. Our two main options here are going to be doing a linear DNA or gene fragment synthesis or gene synthesis. It all goes by different names. It's different names for the same option here. The other option would be to do full synthesis, and this would be doing plasmid synthesis going throughout the entire loop. That means that we're going to be synthesizing ITR, insert, ITR, and backbone. Now, really, you have a lot of options here. Depending upon which way you want to go. We do offer synthesis options, and this is available at both the RUO level as well as the CLIA and GLP synthesis levels. Depending upon the stage that you're at, you may consider any one of these. And again, we know you have a number of different service providers here. Regardless of which one that you pick, you're going to need to move on to the next stage, which is actually sequencing your product. So we want to think about sequencing that full plasmid, or at least the AAV section ITR to ITR. Now, it's really critical to make sure that we sequence this entire section. Ideally, we want to sequence the whole plasmid, but sometimes we might opt to just sequence our the insert of our constructs. However, it's really, again, critical to make sure that we have that ITR intact. Traditional Sanger sequencing approaches really struggle with the ITR region. So depending upon you're familiar with this product, you may or may not have already been doing this approach. At Azenta, we have a proprietary ITR Sanger sequencing protocol where we can actually read straight through this ITR so we can get comprehensive sequencing. And this sequencing is really critical that we continue to check this repeatedly with every product that we develop. So original studies here, we used to use a lot of digestion-based methods. That's sort of the standard approach. And so I'm showing you here a nice case study where we had an example of a sample where we did the digest and we saw the expected pattern indicating that our plasmid was fully intact. However, when we did the AAV ITR Sanger sequencing, we actually saw that the part of the ITR arm was in fact deleted. And so this is just a really nice example of a time where we really do need to go down to the sequence level to make sure that we have this fully intact. Now, as I mentioned, you have the option here of doing traditional Sanger sequencing, which is going to go, you can do just the ITRs, you can do ITR, insert ITR, the choice is yours. But we also do offer high throughput sequencing options. And for this, I highly recommend PacBio. Now, there are actually a number of different next generation sequencing methods that you can use, and we'll get into that in a little bit later. But one thing to really consider here is that when we're thinking about our plasmids, we're going to want to sequence that whole plasmid, including the backbone. This makes sure that we have everything intact, that nothing weird happened during the process to actually perturb the backbone itself. But this also allows us to analyze clonality. Now, again, depending upon our approach here, we may anticipate that we have clonality, but especially if we're having a problem plasmid that's not yielding the results that we expect downstream, it can be a good time to go back and actually confirm your clonality. So with PacBio, we're going to get a continuous single long read for each plasmid that we look at, and we're going to get single molecule resolution. So you can imagine here, we're basically going to take this plasmid, linearize it, sequence it totally end to end, and we're going to get a bunch of sequences. So we can actually confirm the full length sequence and we can get the clonality of the sample. PacBio is really excellent for hard to sequence areas, including GC rich regions and ITRs. So it's really ideal for this approach. Now, what happens if we find that there is something wrong? So let's say that we opted for a synthesis with one of the other guys and we found that we had an error. What can we do? So this is where we may want to consider something like an ITR correction service. If your actual gene insert is looking great, but you're having issues with the ITR on the outside, we do offer an ITR correction service. The other option may be to go back to the drawing board and do a complete plasmid synthesis. Just depends on what's going to make the most sense for your workflow. But again, something to consider that if something does go wrong, there are solutions in place to help correct that. Now, once I have my absolutely perfect plasmid, I've done all of my sequencing, I've done all of my testing, and I can tell that I have a great, intact, fully comprehensive plasmid, the next part is plasmid prep. And with plasmid prep, we really want to make sure that we're using our best foot forward here because we know that these ITR regions are really prone to mutagenesis. So here I'm presenting you a brief case study that we did with our proprietary cell lines. So with this approach here, we compared our 2 proprietary cell lines with other companies' proprietary cell lines. And then we picked several clones from every plate, and we actually looked at the ITR sequencing across the board to see what kind of rates we were getting. So were we seeing mutagenesis, or was everything intact? And we found that we have a 98% success rate on the first attempt for scale up at maintaining that ITR integrity. Again, I can't emphasize this enough, something that we need to consider over and over again when we're looking at these products is that we don't want to just do that sequencing at one stage, but at every stage, because all along the way, we do have the ability to introduce mutations or truncation events that can then impact our final product. Okay, so we've created now our perfect transfer plasmid. We've scaled it up. We've got a whole bunch of it, and now we're ready to do the packaging. For the packaging, you're going to pick your preferred host cell line. This is typically going to be a HEC, but you could use SF9 or something else. And then you're going to introduce that transfer plasmid, your rep cap, and your helper plasmid, and you're going to generate these viral particles. Now, our next step is to actually look at the viral particles to make sure that we know exactly what is in here, and that is what we expect. And so to do this, we're going to do package sequencing of the AAV products. For the first wave, I highly recommend that we start with next generation sequencing as a QC method for this packaged product. And with this type of analysis, we're going to be looking at 3 main factors. First off, purity. And by purity, I mean what is within our AAV particles. In theory, we want only that recombinant AAV genome, but we know that we could have other potential contaminating products. And that can include rep cap or helper plasmid, plasmid backbone from the transfer plasmid, or host cell DNA contamination. The next thing that we're going to want to look at is clonality. Just making sure that within that recombinant AAV genome that everything is the same, that we don't have any point mutations, that we don't have any insertion deletions, that it's fully a fully clonal representation of that recombinant AAV. And then finally, fidelity. And that's going to really focus specifically on that ITR component, making sure that we have a full intact product and that we don't have any truncation events or shorter products present. And so to do this, we're going to do a few different methods. I'm going to take you through this entire workflow step by step so we can just discuss all of the different things that we'll see along the way. One thing to really note here is that even though this is considered sort of one comprehensive workflow, there's a number of different stages, and we're going to learn some really meaningful information along each step of the way that will help to guide our final analytics. So to get started, we're going to start with those AAV particles, and we're going to extract the DNA from it. That means that we're going to take those capsids, we're going to crack it open, and we're going to get everything inside. Now, depending upon what we are doing with that sample upstream, we may also be capturing any residual DNA that is outside of the AAV particles. Depending upon how you want to look at that, if you are interested only with focused on what's inside of the particles, then you would want to do a DNA pretreatment. However, sometimes, especially if we're thinking about a more mature product that's going to be sort of in a later stage, we may also be concerned about any extra particular DNA that's present, in which case we do not want to do a DNA digest. So something to just consider here is that when we're doing this extraction step, we're going to be capturing any DNA that's present, single-stranded or double-stranded, and within or the particles or outside of the particles. Now, once we've extracted that DNA, we're going to be able to do a preliminary QC, and this is just a gel-based image of our product. I'm showing you here 2 different examples of what this may look like. I had so many different examples here to choose from, but here's just 2 really nice types of things that we might consider seeing. So I'm showing you here what I would consider to be a pretty good, maybe not perfect, but pretty good product on the top, and one that's maybe just a little bit less ready for prime time on the bottom. So on the top here, we're going to see on the left-hand side, this is our ladder, and then this is the gel image of our products. And over on the right here, this is a digital readout of the gel. So you'll see here we have a lower and an upper marker. That's going to be these 2 bands right here. Our primary product is right here. That's going to be this peak right here. And then you may be able to see it on your screen. It might be a little difficult, but there's a couple of very faint bands here. And you're going to see these as these other peaks. Now, when we're looking at this, we're going to get a numerical value. This is a digital readout, a digital estimate of what we see here. This is not a hard and fast value. So we're going to take this with a grain of salt, especially considering secondary structure with AAV. We know that we're not going to get an exact measure. But what we should anticipate here is that if we have a very pure product, we will have a primary peak at our given close to genome size. If I see nothing else here, if the rest of this is totally flat, that's going to indicate that I have a perfect pure product. However, as we can see here in this particular example, I see a couple of small peaks down here. Now, these small peaks, I can see what sizes the products are. Generally speaking, again, this is our estimated size. I don't know what they are yet, though. So these could be partially packaged products. These could be alternate products. It could be something part of these contaminating products, again, plasmid or host cell. I don't know that information yet, but at the very least, I can see if I have anything else present and the general sizes. Now, like I said, there's a lot of different, depending upon the stage that we're sequencing this, if it's something that's maybe earlier in development or later in production, we can anticipate that we might see other things present. So on the bottom here is an example of a sample that has, a little bit more happening for it here. So we're still gonna have the upper and lower markers as shown. But what you can see here is that in addition to that primary product, I'm seeing a much taller pattern here. So just a little bit more height under the curve and a number of different peaks and valleys. And that's really indicating that I have a number of different products that are present at a variety of sizes, ranging from very small to larger. So again, when we're looking at this, this is really just going to prime us for looking at our downstream results, but we can tell based off of this if we should anticipate a high level of purity or if we should anticipate there being alternate products present, whether it is a fully alternately packaged contaminating product or if it's just that we may have some partial genomes. Now, once we've completed this QC, like I said, this is really just priming us for this information. So that we have a little bit more intel and have expectations set in advance, but we're still going to go ahead and we're going to get ready to do our library prep and sequencing. Now, before we do that library prep, we're going to make sure that everything that's previously single-stranded is now double-stranded. And so we're going to do either single-strand synthesis or single-strand conversion, and then we're going to complete our library prep. To do this, we are going to look at two different methods side by side. We're actually going to be using both of these. So for a single sample, I would do a single extraction, and I'm going to actually split the DNA at this stage and do both of these options at the same time. So with our second strand synthesis library preparation, we're going to do Illumina short read sequencing. That means that we're going to be sequencing very small fragments, and then we're going to map those back. These are going to be 150 base pair paired end reads, so 300 base pair pieces basically. And we're going to be using an amplification-based approach with this. What that means is that we don't need a lot of input, but it also means that we're doing very short sequencing and we're going to do it very deep. So when we look at these results, we want to think of little Lego blocks that we're going to be stacking all up, mapping back to all of our different potential genomes. I'm going to get into what that means for our analysis in just a second. Our other option here is going to be PacBio long read sequencing. And with the PacBio approach, we're actually going to take all of these DNAs, we're going to do a second strand conversion, and then we're going to do a library preparation that does not involve amplification. We're just going to be adding these SMRTbell adapters as shown onto the ends, and we're going to be sequencing all of the molecules end to end. That means all of our full length products as well as any smaller products that might be present. And we're going to be doing a still deep, but not quite as deep sequencing with PacBio. So when we're thinking about these two options, we really want to think about Illumina as being short, but very deep, and PacBio being long and intact, slightly less depth. And so if we think about our analysis here, we're going to get a really comprehensive view of what we're going to see with these two different technologies, and we're going to see how they interact with one another to give us a holistic, comprehensive view. So again, remember, we're going to be thinking about our purity, our clonality, and our fidelity. Those are our three main things that we're concerned about. So thinking first about the purity. With our Illumina sequencing, again, I'm taking those very short blocks of DNA, sequencing very, very, very deep, and then I'm going to map all of these sequencing reads back to anything that it could be. I'm going to map it first to my recombinant AAV genome. That's going to be my ITR, insert ITR, potentially backbone, rep cap and helper plasmid, and the host cell line. I'm going to do all of this mapping, and I'll be able to get a really, really excellent view of what frequency is recombinant AAV and what frequency of those reads is anything else. There's really no bias associated with this method, so it's going to give me a very accurate result with regards to the frequency or the purity of the sample. We can detect contaminants well under the 1%, even under the 0.1%. So we'll have a really nice view of, do I have a very pure product, or am I seeing other sequences present that might be contained within those viral particles. Once we've confirmed our purity component from that step, the next part is going to be thinking about that clonality. And again, clonality is really thinking about what is the sequence of my AAV? Is there, do I have any point mutations or insertion deletions present? And if so, at what frequency? Again, we want it to be totally clonal or no changes. With that Illumina sequencing, we're going to do those short reads. We're going to look at just the AAV now, ITR to ITR, and we're going to stack all those reads, and we'll get a really great mutation frequency for our samples. And this will let us calculate any abundance of variants that might be present. Again, very accurate, and we can detect well under the 0.1%. I'm going to show us some example results here in just a moment. Now, once we have our Illumina data processed here, we're going to look at our PacBio data. Again, these are actually both happening simultaneously at the same time, so we're really looking at all of these results at once. But once we look at our PacBio long read sequencing, we're going to get more information again about our clonality and fidelity, but we're also going to get insights about the purity. So thinking about that recombinant AAV genome first, with the PacBio sequencing, again, we did that long read end to end. So we're going to get the full length recombinant AAV genome integrity. That means that we're going to take all of those reads and stack them up, and we'll be able to see everything from ITR to ITR, and we'll have full ITR coverage. That allows us to really make sure that we have complete intact genomes. How much truncation do we see? Do we have any truncation hotspots? The next thing that we can look at is within that entire product, do I have any point mutations here? Not specifically the truncation events, but do I have anything else that's happening here? So I can have a mutation event again within the gene insert, but also within the ITRs. So we're really going to reiterate what we saw previously in the Illumina data, but we're going to do it now in the context of that full-length molecule. And so with the Illumina and PacBio together at this stage, we really have confirmed or validated everything that we're going to want with regards to the clonality and fidelity. Now, coming back to that purity component, we saw with our Illumina data that we may have other species that are present or other types of DNAs that are present. With the PacBio sequencing, we're going to actually be able to resolve what that looked like within the molecule. So if you'll think back to that gel image where we saw additional peaks present, we will be able to see if I had, say, for example, some host cell DNA that was contaminating, I will see how much I have, the frequency with my Illumina. I'll see where it has mapped to with Illumina. But with PacBio, I'll be able to resolve exactly what that piece of DNA looks like within the particle. Was it big? Was it small? You know, what did I have a chimeric event? What did it actually look like when we were in that packaged product? And so if I put both of these analyses together, I'm going to have a very comprehensive view of exactly what my AAV sample looks like. Now, I want to take you just through here a couple of example images here. We do have a number of, we have two main reports for these. And after the webinar, if you do have any questions, please feel free to reach out and we can actually send you over example reports as well. But for our reporting here, we're going to be able to actually visualize all of those things that I just discussed. So at the top left-hand side, we have our variant calling here. So you can actually see the frequency, and you can see here my variant frequency is quite low, but we can actually see presence or absence of any particular point mutations. At the bottom panel here on the left-hand side, we're going to see if we have any truncation events, and we'll be able to actually identify truncation events within the transgene and within the ITR. You'll always notice with these images that the first and last nucleotide will show like a slight peak, and that's really just showing us where we have our ends of the product. With our Illumina data, we're going to have a read mapping for that contaminant quantification. So here I'm showing you an example report where we had a reasonable purity, but we did see a pretty fair amount of RepCap and helper contaminating products. You'll get a similar set of content from the PacBio, but due to some size bias, we do anticipate that that's going to be less accurate. So we really want to rely on Illumina data here for giving us those best stats for this particular product. However, again, when we think about that end-to-end sequencing with PacBio, we're going to be able to really resolve what those products look like. So in this particular example sample, you're going to see on the top that we had a pretty high amount of that RepCap contaminating plasmid within the product. And if I actually go down here into the chimeras, you can see that we're actually getting a large number of chimeras between the AAV genome and RepCap. So it's kind of reiterating what we're already expecting here with this particular sample, that not only are we seeing that high contamination, but that it's having an impact on our AAV in this particular example. So holistically, we're going to take those two methods along with that primary QC, and we're going to get a really great view of what's happening inside of those viral particles. Now, this is just one example of a sample, but hopefully once you're going through your entire workflow, you're going to end up with a perfect, excellent AAV that you're going to say is now ready for prime time. And once you have that ready, we strongly recommend that you consider doing a GLP AAV ITR Sanger sequencing. This is a regulated version of Sanger sequencing that you can use on your final product. So we won't be looking at the contamination component, but really focusing on what is our AAV sequence. This is going to be done with QMS oversight, and we're going to be going ITR to ITR. So Sanger sequencing all the way through across the entire packaged product. And again, this is in a regulatory environment, which makes it really ideal for your FDA IND filing. Now, moving ahead through this development workflow, I took us through that sort of entire process, and you can imagine that depending upon where we are in those stages, we're going to actually be straddling both the discovery and preclinical. Within this preclinical workflow here, when we're thinking about our model development, yes, we started at lead identification, but we're going to be continuing those same tools as we start to refine our product. So this is where we're going to start characterizing those targets by selectivity and potency, performing animal studies and toxicology studies. Again, we have a number of different products and solutions here within the genomics and analytical services, but we also offer a number of different consumables and instruments if you decide to do that in-house. And then finally, really preparing everything for the regulatory and FDA review. And again, with these parts, once we've reached this stage that we're ready to start creating our filings and getting all this paperwork together, that's where we really want to consider moving into regulatory services. We may be doing a mix and match scenario of both RUO and GLP, but we do have a number of GLP services that can really complement this workflow. And at this stage, this is also where we're going to start thinking more seriously about our storage options. So previously, we might have been storing all of our stuff within a freezer on site. Obviously, we want to make sure we're using that great freezer, not the one that beeps all the time. But we also might want to consider at this stage, once we're getting further through our preclinical development, off-site storage. Off-site storage is really critical because it's going to allow us to have backup of our products, especially as we're starting to get more mature. We're going to want to have those backups available just in case. But also, as we're starting to expand our animal studies, we may decide that we want to save some of those animal samples also off-site, both as backup and because we may not have enough space to actually handle the number of samples we're receiving at that stage. And so for this, we do offer off-site storage that's regulated storage. So depending upon your storage needs, a lot of options here. Okay, now we're getting into the last stage, clinical development and use. And so this is where we're really thinking about the clinical trials. Now at this point, that means that we're transitioning from animals into people. And we're going to start out first with the safety studies and then with the efficacy studies. And then we're really thinking about CLIA level services. Now, depending upon what we're looking at here, we're going to be thinking about next generation sequencing, Sanger sequencing, proteomics, qPCR, a whole host of different assays. And some of these are going to be related to the therapeutic itself. Some of it is going to be related more to patient stratification. As we're doing this, we're going to be using a number of different genomics and analytical services. Depending upon how that's going, we can also assist with enrollments. Now, once we have this clinical trial going, we're again going to be thinking about storage now, but storage of those human samples, right? All of the patients who are enrolled in this clinical trial are going to have a number of different samples that might be collected at different time points. And so we're going to want to make sure that we are storing all of these products safely for future reference, but also just with regards to requirements by law. We're also going to be thinking again about that storage of the drug products, but also distribution of the drug products. And so to speak to this a little bit further, Azenta does have a number of different clinical trial solutions that are specifically focused just for clinical trials. So this includes clinical trial sample management and really just thinking about organizing your samples, gathering samples, and analyzing samples. Patient safety testing, drug product storage and logistics, that's really thinking now not on the patient samples, but on the products itself, and including cold chain storage and management. And then finally, informatics solutions. Informatics solutions is going to be really critical at this stage because as we start gathering more and more individuals, we're going to have more and more data. And I'm talking about the informatics solutions here, but we may even consider bringing those in during our preclinical stages because we can actually use the same tool with our animal studies as we're starting to gather data there, depending upon how we want to actually organize our results, but also to just help us track this information. So while I have featured it here, we can actually even introduce this solution much earlier as needed. Now, we focused really heavily a lot on the drug product, so I did want to also talk about manufacturing and scale up. So as we're really expanding that clinical trial, we're thinking we're going bigger, we have more patients, we have more need. We really want to also start thinking about the storage and cold storage logistics. Really, really critical because we know that depending upon the product that we have, this can be very temperature sensitive and that can have huge implications on stability, also transport. So we do offer cryogenic storage systems, we offer logistics systems, and then we also have our cryopod, which is a really nice option for actually transferring samples between different pieces of equipment. As I mentioned, we also have other types of logistics in the broader spectrum. So the previous logistics would be your on-site logistics, but we also offer off-site cold storage, cold chain logistics, and then inventory management. Again, when we're thinking about these clinical trials, we want to be thinking about not just our human subjects, but also the actual drug products. And so if we put this all together with our entire gene therapy development workflow, we actually are, I hope you've seen today that we have a number of different products and solutions under the Azenta roof that really are going to take us through this entire process. So starting with that early R&D, target discovery, lead identification, model development. And in that stage, that's where we're going to really think about actually creating that AAV and working on all the different things that we need to think about. Process optimization, where we're going to actually start looking more at our animal studies in the preclinical stage, making sure that we're hitting that safety and efficacy within the animal models, and then carrying it through into the clinical trials, therapeutic delivery, and scale up when we're bringing this out into the human population. And so across the board, we're going to be discussing our genomics and analytics, consumables and instruments, data and informatics. That includes those LIMS systems, sample sourcing, storage, automation, and logistics, both on-site and off-site, and then any consultation. And one of the big perks about using Azenta is that because we offer all of this under one roof, we can really streamline this approach to you to help you get everything to market faster. Now we know that you guys have a lot of different service providers for all of these different solutions. One thing that's really great about Azenta here is that we have a lot of experience within this space. So I'm just showing you here some of our next generation sequencing stats in particular, but we do offer some of the highest on-time delivery rates. We have a ton of experience in this space. We're an accredited lab with regulatory oversight as needed, and we offer stellar PhD level support. And so with that, I thank you all for listening, and we're going to get ready with our roundtable discussion featuring a panel of speakers. And so if I can please have my roundtable friends join me on camera, I'm going to go ahead and turn our slideshow off.
Thanks, Andrea. Great job.
Thank you.
Perfect. Thank you, everyone. So we're going to move to the roundtable now. Just to orient everyone in the audience, there's a little drop-down arrow that says questions. If you click that drop down arrow on the right, that's where you can add questions and we'll see them come through. But while you guys are drafting your questions, we'll go ahead and introduce the roundtable. So could the roundtable please go around and introduce themselves? And Andrea, could you start us off and reintroduce yourself? Thanks.
Sure. So I'm Andrea O'Hara. I am one of our strategic technical specialists, and I focus on next generation sequencing, but also our cross Azenta solutions. And I am a specialist for our cell and gene therapy space. And I'm going to go to Liz L next.
Yeah, I'm going to go. So I'm Elizabeth Louie. I'm the supervisor of the technical applications team for the genomic side of Azenta. Our team focuses on pretty much all of the genomic solutions. So a big focus on NGS, but any of our synthesis and prep and other solutions that we offer. So I've been with the company for about 10 years now, and I have a PhD in molecular and cellular pharmacology. And I will pass this to Candy.
Hello everyone, this is Candy Mao, and I am a bioinformatics scientist in Azenta. So our group focuses on data analysis and pipeline development for different pipelines. And for me, I joined Azenta at the end of 2020. So it's a little bit, almost 1.5 years. And I have a PhD in biochemistry from Purdue University. I will pass to Ming.
Hi everyone, my name is Ming Yang. I'm a business process manager under the gene synthesis department at Azenta. I work within different teams within the business to support operation and business growth, and also cross-functionally with other teams within the company. I have a PhD degree in molecular and cellular biology, and I'm happy to be a panelist, answer any of your questions about our services. I'll pass it to Liz?
The other Liz. Hi, so I'm Liz Zhang. I'm an associate director in applications product marketing at PacBio. So I'm very happy that you guys invited me. It seems like everybody talks about the PhD, which is a really long time ago for me, but I have a PhD in computer science from the University of Washington.
Fantastic, thanks team. Okay, so we'll go ahead and start answering questions. Just to start us off, Ming, we have a question here for you. What types of QC do you have for AAV synthesis and plasmid prep, and why do you think those are important? I know we went through them in the talk, but it looks like there's some more interest there as well.
Yeah, thank you for the question. It's definitely a great question. So for plasmid construct generated from our AAV synthesis service, the QC actually includes all the steps of our standard gene synthesis service, including full Sanger sequence verification of the synthetic insert, as well as restriction digestion of the final plasmid to verify the overall integrity of the final construct. And in addition, for our AAV synthesis plasmid construct, we actually also perform ITR sequencing in the final plasmid preparation, which will be delivered to our customers to ensure that we have 100% correct ITR regions, which are important for the downstream AAV packaging. So those steps are very important in terms of confirming the sequence accuracy of your gene of interest, and of course, as I mentioned, the integrity accuracy of the ITR regions, and all of this tied together to make sure that the AAV plasmid construct generated from this service will help your downstream application to generate high quality AAV viral particles, enabling your research. Hopefully that answers the question.
Perfect. And then one that might go to you as well, Ming, but just came in. Can you address any differences between cell lines HEK293 and SF9 cells? Any differences or benefits or trade-offs there? And that can be for the whole team too.
I'm happy to actually take that one. So the HEK293 is a pretty standard model. That's going to be human cell lines. SF9 is actually an insect cell line. So the standard workflow here is typically going to be human cells. However, some researchers or companies might opt for the SF9, with the idea being that if there was, I'm going to use air quotes here, host cell contamination because it's an insect cell line, that it wouldn't be things that are going to be compatible with the human cell line. So if something else was packaged, it would not be oncogenic. That's sort of the goal in that approach. So that's sort of one reason why we might want to consider an alternate model. I would say that that does tend to be a little bit less typical, and that more often than not, we're going to stick with the HEC 293 because this is a very robust system.
Just to quickly add on to what Andrea has mentioned, in terms of the SP9, which is the insect cell line, we do see more applications, for example, protein expression actually using baculovirus instead of adeno-associated virus. We know, our seminar is focusing on adeno-associated virus, but, just want to have a quick comment there in terms of baculovirus used in insect cell in terms of protein expression. And we actually do offer baculovirus service. And just a quick comment, if you're interested, feel free to include that in the survey question feedback that we're going to send after this webinar. Thank you.
Awesome, great. Let me scroll here, sorry. So Liz L, I think this would probably be a good one for you. For NGS sequencing and for AAV QC, why do you use amplification for Illumina?
Yeah, so I think that's a great question. So it's kind of just based on what the goal of that project is, right? Or that goal of that platform. It's really to be able to detect anything and everything that's in that sample, whether it be your AAV or potential contaminants. And it's just a limitation of that library preparation that does require an amplification step for either the second strand synthesis or the second strand generation, so that we can detect everything and anything that could be in there.
Perfect. And then maybe to build on that too, Liz, this is kind of a question about some of the slides that we presented, talking about how PacBio can sequence from ITR to ITR, so Liz T. So how does that benefit the AAV process from a PacBio perspective?
Yeah, I think Andrea also talked about, I think when you see the gel, it's really obvious that, if everything goes well, you have a peak, you're probably done. But the fact that you can see non-full length, because you're sequencing full length, I think the word full length is interesting because we're sequencing, it's faithfully sequencing whatever you put it. And if they are non-full length, the AAV that's being packaged, you will see it as in, I think we should use the word intact, which I know, Andrea, you were using, is that we can see the intact AAV. We know we didn't fragment it. We know we also didn't amplify it, so that if there are truncation events, which there's actually quite a few publications, just Google PacBio AAV, you'll see that these do actually happen. And then in this case, you'll be able to see that really clearly.
Awesome, perfect. And then another question, I believe about plasmid, but I'll open it to the whole team here. How do you check to get high purity and high yield? And can you kind of explain what that looks like? We can pass that to Ming if you want.
Yeah, I guess I can comment in the context of AAV plasmid synthesis. So, so after synthesis and cloning into AAV backbone plasmid, the correct clone is actually, picked from a single colony. So at that step, it is a clonal product. And of course, we will proceed with our sequence verification using our proprietary AAV ITR Sanger sequencing, as well as regular sequence sequencing across the synthetic insert. And all of this, the actual peaks that you actually observe in the chromatograms will actually have a pretty good indication of whether we have a pure and clonal product. And then when we move on to the plasmid preparation, and for example, we actually need to scale up to a larger scale DNA plasmid preparation. We actually also sequence verify the junction of the synthetic inserts after the large prep, and also, of course, the ITR regions. And so all of this also, will make sure that the final deliverables are clonal, purified, and also sequence verified plasmid constructs.
Yeah, and I just want to add in on to Ming's, if purity is a concern at the plasmid level, I think Andrea mentioned that PacBio was an option for sequencing to really look at low-level contaminants that might not be captured with Sanger. For example, if you're worried that there could be something that isn't your plasmid backbone at all, but something completely different, then using the NGS route, very similar to doing like the AAV genome work, is definitely applicable here.
And then one more thing just to add, on the actual panel here, if you guys have seen it, there is a handout section We actually do include some number of content and tech notes regarding these different processes. We do also have some really excellent white papers here. So after the webinar is over, you will have an option to include some comments. Please let us know if you need any additional literature about any of these products or services.
Yeah, great, awesome. And then it looks like there's some interest in speed. So can we talk a little bit about how quickly we can do PacBio ITR plasmids for AAV? What's the turnaround time there?
I'm happy to take that one. So we actually offer a number of different turnaround time options. So for the final packaged product, we're typically looking at 3 to 4 or 4 to 6 weeks. That's going to be submission of viral particles all the way through to analysis. Similar turnaround times for plasmids. Again, you can think about an expedited option in that 3 to 4 week range, or a standard turnaround time of 4 to 6 weeks. If you ever need something faster still, please do let us know. We do have what we call lightning speed, which is going to be extra fast options. So just let us know if you ever have a firm deadline that you need something to be processed with a little bit more speed and care.
I'm going to jump in again, sorry. So if you do need something faster and you know that you might have a clonal product, we also have options for kind of what we term like sequence confirmation using the Sanger, especially if you're looking at the plasmids. So if you're not necessarily interested in the contaminants, that requires PacBio, and Sanger is an option. And we can do all of the primer design and all of the analysis for you here as well. That typically takes about 1 to 2 weeks, just really depends on what information you have and how big that initial plasmid is.
Thank you. Okay, and then we had a question for Andrea and probably the team as well. So Andrea mentioned that there was a size bias with PacBio. Is there anything you're working on to understand or resolve this?
Yep, that's a great question. This is correct. There is a size bias, which basically means when we were looking at that gel image for the that primary QC, remember we saw the one that had a lot of other products at the bottom. What you kind of want to consider here is that if we looked at that area under the curve for the smaller products, after we've done the library prep and sequencing, we would anticipate seeing a little bit more sequence represented for smaller products versus the larger ones. Now, to get around that, we do have an option that's going to be a lambda phage digest spike-in. This is basically adding a ladder to your sample that you can use to normalize the results. And I'm actually going to let Candy comment here when I'm done, because she has assisted with the analysis of this portion. But it's a really great option for helping to give us more frequency resolution for those samples. However, it's most ideal for samples that do contain some level of contamination and not a sample that's totally pure. So we really want to use that as a consideration for those samples that are maybe showing us that there are other products. Candy, anything to add?
Yeah, thanks, Andrea. I think that's very nice for the explanation of the lambda spike-in. And yeah, we do add lambda spike-in and the correction in our data analysis. And so far, as we understand, different projects are different, different products are different. So we actually have lambda spike-in analysis as well as the regular analysis if we do have lambda spike-in. So you can, for our audience and our customers, you can compare both results and pick the one that you think that's most reasonable for you.
Right, and that's a really important option, a really important comment, that you're going to get that dual analysis. So if you opt for spike-in and it did turn out that you had one of these very, very pure products, you may opt for the non-spike-in analysis, as that will actually give you more representative data. Again, when we're thinking about that frequency information here, we're still going to rely heavily on Illumina, but that's going to really help us to resolve the frequency of particular sized products.
Perfect. And then I think so a PacBio question here. For PacBio, does second strand synthesis or rehybridization of extracted single-stranded DNA viral genomes miss shorter genomes because of repair? Liz T. We can take that one offline too, but I thought I'd pitch that to you.
Yeah, well, I think this seems to be a question both for Azenta and for PacBio, right? Because it depends on what the second strand synthesis approach is, but I think that's a good question I want to bring back. One thing I would say is that PacBio does have a library protocol that I think is similar to Azenta's, and that's in the handout. But one of the drawbacks of us working on this protocol is, well, I shouldn't say drawback, is we don't have as much real world samples to work on. And so when we tested it on our data set, which is actually in the PDF, we can't see anything that's not full length. If that makes it sound kind of sad, is that we actually have too good of a pure product to work on when we develop the protocol. So I would be interested, you know, if anybody wants to work in a more collaborative research space with us, or you think you have shorter, truncated, or contaminated products that you want us to test it out, we'd be happy to.
Perfect. Any other comments or questions on that one? Okay. And then it looks like we had some interest just in BI. So Candy, maybe you want to help with this one, and the whole team as well. So what methods do you suggest for identifying unknowns in your sequencing data? What tools should you use to identify what those are, and how can you, kind of use that to your advantage?
I can start first from the BI aspect. So I guess the unknown will be like unaligned sequences or something, but it could be something else. But for the unaligned sequences, we do provide alignment output, so you can actually get the sequence from that output and then do some bioinformatics tools, like using some bioinformatics tool like BLAST or something else to figure out what they are. And for if if you have specific how to say the precaution of what may be this contaminate, then you can also provide us the sequence of that contaminate ahead of time, and during the alignment, we can include that as one component similar to RepCap or helper or other things, and then we can output the alignment details for that part as well. So that's my understanding for this.
Perfect, yeah. And I think we're coming to the end here, so I'll just add on to this question because it's kind of a hybrid of what we were just talking about. So there's a question around, can you use the AAV NGS solution to look for contaminants across your process flow? So where you're actually looking in your manufacturing stream, can you use that tool across those samples? And I'll open that to whoever wants to answer here.
I can go ahead and get started with that one. Yes, so as Liz L already mentioned, that Illumina system is really meant for us to see anything and everything that's in the sample. So it's really ideal for identifying if there is anything else that's present in there. And that could, again, in this perfect workflow that we have, we really only expect there to be certain products. But if we were concerned there was something else there, or if for some reason we had introduced something else into that sample, a good example might be if we were doing something where we had maybe multiple products prepped together for some reason, that we would be able to see all of that at once. Illumina sequencing, because it is really unbiased with all of that, we're going to get a chance to see everything that's in there. So a really good ideal workflow for that. And as Candy mentioned, depending upon how that analysis is processed, if you have specific things that you anticipate being in there, that might be in addition to the standard. We can actually specifically search for those. If you're more concerned about what else is in there that I didn't think about, there is going to be actually a set of reads that are going to be called unmapped reads, meaning they're everything that didn't go to our known references, and we can actually take those, blast those to see what they are. So there's a lot of options at our disposal depending upon how thorough we want to be and what we're all looking for.
Perfect, thank you. That was good for our last question. So that's all we have time for today. If we weren't able to answer your question in the chat, we'll follow up with you via email. We do have a 5-question survey at the end, and your feedback would be great for us to learn as we kind of build more of these workshop events. Once again, we'd like to thank Dr. O'Hara for her presentation and our whole round table of speakers. Thank you for all the great insights. And thanks to all the attendees who attended today. If you'd like to view the workshop, we will make it as a recording, and then we can make that available to you shortly. So again, thank you, team, and have a good rest of the day.
Thank you, everyone.
Thank you. Bye.